This is the second half of the evidence lecture. Your class might have gotten a little bit farther than this in class, so you can fast forward to get to the point where we left off. We left off in some classes on analogous structures that are caused by convergent evolution, which is evolution towards similar traits but in distantly related or unrelated species due to similar selective pressures in their environment. We use the example of a shark and a dolphin that live in the same environment and have similar body shapes, fin placement. They eat similar foods. However, they are not closely related at all. A dolphin is a mammal and a shark is a fish. So, but they do look alike. That's not to mean that they are closely related just because they share those traits. Vestigial structures are structures that are remnants of an organism's evolutionary past, but currently have no function. So these are things that have been passed down from a previous ancestor that used these structures, and now they're no longer useful. An example would be the wings of an ostrich. It doesn't fly, but its ancestors would have flown and hence used those wings. The eyes of a mole, even though the skin and fur covers the eye sockets, there are still eyes underneath that skin. They don't work, so even if they were to injure themselves and have that skin ripped open, you would not be able to see with those eyes, but they are there, and the eye sockets are there because an ancestor of theirs did have eyes that were able to see. Both whales and snakes have a vestigial pelvis, so this tells us that they must have had an ancestor that had legs, hind limbs, because the pelvis's only function is to connect the hind limbs to the backbone. So this structure would only be needed if that organism had hind limbs. Obviously, snakes and whales no longer have them, but they have the pelvis left over to show that they that their ancestors did. This is an abbreviated list of some of the vestigial structures that we find in humans. If you would like to take a look at some of these that are kind of interesting, you can pause it here in the video and take a look at some of those. Feel free to email me with any questions that you might have as well. This organizer is found in the note guide that is also here on my website. So if these are helpful to you, you may print this and fill it in as we go through it right now. The last portion the last piece of evidence for evolution is DNA and proteins. It was too large to fit on the other concept map, so it has one all by itself. So what we're looking at is evolution today really unites all biological fields. It is a core concept that is between many different subject areas in biology. It's supported by paleontology, which we've already talked about, and supported by molecular evidence. Molecular evidence is what the rest of this lecture is about today. For molecular evidence, we look at protein comparisons. We know from our genetics unit that proteins are built based on the genetic sequence in the DNA, so protein differences are akin to DNA differences as well. So protein differences, by looking at those, we can create a molecular fingerprint, which we'll talk about later in the lecture. Molecular evidence also shows probable evolutionary relationships, and we often see them confirmed with DNA from the other types of evidence that we've seen already in the lecture. That should say species, not spices. For this is also supported by genetic evidence, which is the same as molecular evidence. We see this in pseudogenes. Pseudogenes are genes that do not code for anything. They reflect a common ancestor because they are similar among many different species, but they don't have any use. They are heritable, so they're copied and passed down during DNA replication. But they're vestigial, meaning that they don't actually serve any purpose in today's DNA sequence. We'll also talk about homeobox gene clusters, which are also called Hox genes. So molecular biology, we look at similarities and differences in DNA and proteins. We often look at things that many organisms have, such as hemoglobin or cytochrome C, which is another protein. These are going along with the theory of evolution, closely related species will show more similarities in these molecules, and distantly related species will have more differences. That meaning that the longer ago two species diverged, then they will have more time to accumulate differences than two species that diverged a more recent time ago. So we can make molecular clocks, which, based on how long ago species diverged, we can tell how long ago it was based on the number of differences in their DNA or in their proteins. The greater the divergence, the greater the percentage of the differences in comparative molecules. So, for example, we look at cytochrome C. This is a very common protein that we use to compare between organisms. When we compare a human and a yeast, there are 66 differences in this gene. But when we compare a human and a pig, there's only 13 differences. Another one that we often use is the number of amino acids that differ in hemoglobin. The total chain is 146 amino acids. So between a human and a rhesus monkey, there's only 8 differences out of that 146. But between a human and a lamprey, which is a jawless fish, there are 125 differences. So there's a much greater difference between the fish and the human than between the fish, or between the monkey and the human. We can also calculate what genes will accumulate more alterations in the sequence over time and determine the rate of evolution. So that's why it's called a molecular clock, because we can measure how fast the evolution is happening. All right, there's 2 specific types of genes that you're going to need to know, the pseudogenes and Hox genes. First, pseudogenes are sequences that do not code for anything. So these are non-functioning sections of DNA. We learned about that a little bit when we did protein synthesis. So if you think back to that, how would that be related to the RNA processing? Hox genes also come from a very distant common ancestor. These genes determine head-to-tail development in embryos. So even insects to humans, the cells that are going to be at the top are the ones that are for the head, brain area. Cells at the bottom are for your extremities and your tailbone. We see this across all animals. So these genes are shared by all organisms. They're sometimes called master genes because this is a key gene in determining how organisms are going to develop. There's sometimes more than one, but they all are based on the placement of those cells. So when the sperm and egg combine to make a zygote, that's just a single cell that then divides through mitosis to create essentially a lump of cells. Then depending on the location of the cells within that lump, that determines what type of cell they will turn into, whether they'll turn into a brain cell or a skin cell or an organ cell or somewhere down on your extremities. So the position of the cell is what causes the differentiation. It creates segments of cells in the embryos that develop into certain tissues or structures. So it kind of divides up that embryo that will then, those cells are developing into a particular type of tissue or structure. We see this across all animals. This is a defining feature in animals. To be in the kingdom Animalia, all of them have these Hox genes. So even organisms that are more complex or more simple, they all have that gene determining which end is going to be the head, where is going to be the middle, what's going to be the tail. Okay. For the last part of this lecture, I just want to point out a couple of misconceptions that many people have. First, when we're talking about evolution, that does not at all include the origin of life. So evolution is about what happens once life is already here. This does not include how life began in the first place or anything about the Big Bang. It's just about what happened once life was already here. So don't confuse the origin of life with the theory of evolution. Second, humans share a common ancestor with many organisms, but in no way did humans come from monkeys. The theory of evolution does not say that humans come from monkeys. That's not at all what it's saying. In the theory of evolution, we share a common ancestor, but just like is shown in the lab, that common ancestor was neither a monkey nor a human. There's just shared some characteristics. The saying that humans came from monkeys is not is a very common misconception. To say that if humans evolved from monkeys, then why are there still monkeys, is like saying if Americans came from Europeans, then why are there still Europeans? We didn't come from Europeans, we share common ancestors with them and have diverged from them over time. That is the end of this lecture. So if you have any questions, please email me or bring them to class so that we can discuss them, make sure that you have all of your notes filled out, and that you are ready for class on Monday. Have a great weekend.